My name is Ennis, and I'm the co-founder and chief operating officer at Freenome. Freenome's focused on systematizing the early detection of cancer, as well as identifying what treatments will work best for each individual. Our goal is actionability, and it's to leverage not just AI, but also underlying wet lab technologies and platforms, and bring the best analytes and the best information together to answer key clinical questions that are going to maximize the improvement in treatment and detection pretty much across the entire value chain. Today with QIAGEN, we're primarily focused on the initial upfront sample processing, where some of the analytes we look at really benefit from the PAXgene tube and what QIAGEN has really done with the tube innovations. And I would say that primarily in some of the extraction chemistries, that there's certain kits and reagents that have worked really nicely for us. And I think as we look at running some of these tests at scale, there's some additional products that we're thinking about incorporating into that workflow. In the entire workflow of our lab, whether it's on the tubes that might be used, whether it's how we think about some of the extraction workflows for some of these analytes, or even thinking more about automation, pretty much across the entire touch point of this multi-analyte wet lab process, we see several QIAGEN products along the way. And I think technically that's where you align closely with a company that doesn't just think commercially and has a big vision for the impact you can have, but understands technically how do you operationalize that into a clinical test. And that's where a lot of the technology that QIAGEN has really does integrate across the entire process that we've built out at Freenome. Freenome kind of came from caring deeply about the problem in oncology. I had spent some time more on the therapeutic side, and I was always frustrated by the difficulty of initiating an immune response or any durable clinical benefit from late-stage disease. And one of the root causes of the problem was being able to identify cancer at the right time and make sure that each tumor was treated in a way that was matched to the treatment. A lot of the circulating markers come from the immune system and the body's response to the presence of cancer, which is why immuno-oncology tends to be a major focus of the work that we do and plan to do with QIAGEN, because of not just the level of success and excitement in immuno-oncology in terms of improving overall survival, but also that the circulating markers in periphery are coming from the immune system. As we started to apply and develop our own technology platform, where we take multiple circulating analytes in blood and we leverage the AI platform to discover the best biomarkers for that clinical question, we saw that to scale this and have the global reach and the impact, which can be everything from the turnaround time and the level of access to any technology that we can develop, that QIAGEN fit nicely. With our relationship with QIAGEN, the next step for us is taking some of the key partnerships that we have. We've been working with about 7 biopharma companies to date and really hone in on what are the key specifications, both in the US and globally, that will be required for a global launch. What are the key clinical questions and use cases where our current partners are most interested, and how can QIAGEN and Freenome both impact those relationships and move towards the initial proof of concept in this alliance, that together we really do have a multiplying effect of the impact that we can have.
QIAGEN.
Sample to insight.